Hello students, let us start with chapter 2, Nutrition in Animals. First of all, what is nutrition in animals? Nutrition in animals include nutrient requirement, mode of its intake and its utilization in the body. Next, what is digestion? Breaking down of complex food into simple substances. Let us now discuss the different modes of intake of food. Different animals have different methods of taking in food. Let us discuss a few. First of all, the hummingbird and the bee. They suck nectar from flowers. The mosquitoes and lice, they suck blood from humans. The infants in human suck milk from their mother. The python, it swallows the prey it has captured. The eagle, it captures the prey and then swallows it. The housefly uses a method known as sponging, while the butterfly uses the method known as siphoning. And for this, it uses the mouth part known as proboscis. In case of snails, they use a method known as scraping, while ants use the method known as chewing. These are a few in the big list. Now, in case of starfish, which is quite interesting, the starfish, when it comes in contact with its prey, it especially likes to feed on hard shelled animals, and the shell is made of calcium carbonate. It breaks open the shell, and then once that is done, its stomach pops out of the mouth and feeds on the soft flesh of the animals. And once it has fed on the animal, the stomach goes back through the mouth and the food is digested. In the process of nutrition, there are 5 major steps. They are ingestion, digestion, absorption, assimilation, and egestion. Let us see one by one. Ingestion means taking in of food. This is followed by digestion. I have already told you the definition, that is breaking down of complex food into simple. This is followed by absorption. That means the digested food is absorbed into the body. Absorption is followed by assimilation. That means the digested food is then taken to different parts of the body and different organs, where it is converted to complex substances such as proteins. And it is used for the growth and development of the body. And the last one is egestion. That means the waste that is produced is thrown out. So these are the 5 steps. Now let us look into the details of the digestive system. In the digestive system, there runs a long tube which is known as the alimentary canal. The alimentary canal starts with the mouth and ends in the anus. It is also known as the gut. Now the different parts of the alimentary canal are the buccal cavity, the food pipe or esophagus, the stomach, small intestine, large intestine, rectum and anus. Now along with this are found certain glands such as the salivary glands, liver, pancreas, gastric glands and the intestinal glands. These glands produce digestive juices which also help in the digestion of food. That means breaking complex substance into simple substances. Let us start off with the buccal cavity. There are three important parts we have to learn about in the buccal cavity. First, the teeth. In humans, we have two sets of teeth. When a child is infant, he or she starts growing teeth, and this teeth lasts till the age of 8. And this first set of teeth is known as milk teeth. It falls off by the age of 8. Later, new set of teeth grows and it remains in the individual's body throughout his or her lifetime. This set is known as the permanent set. In human beings, we have 4 different types of teeth with different functions. First of all, the incisors. The incisors help in biting and cutting. The canines help in piercing and tearing, while the premolars and molars help in grinding and chewing the food. In the lower jaw is present 4 incisors, and in the upper jaw also there are 4 incisors. The canines are 2 in the lower jaw and 2 in the upper jaw. The premolars 4 in the lower jaw and 4 in the upper jaw, while the molars 6 are present in the lower jaw and 6 in the upper jaw. So there are 8 incisors, 4 canines, 8 premolars and 12 molars. Therefore, a total of 32 teeth. In human beings, we have a total of 32 teeth. Now, what is tooth decay? Normally, we have some kind of bacteria in our mouth, but that does not harm us in any way. But if we do not brush our teeth properly or keep our mouth clean, this bacteria, it acts upon the food that is left over and produces acids. These acids then act on the teeth and cause toothache, and also it destroys the enamel of the teeth. This is known as tooth decay. Now, how can we avoid tooth decay? We need to maintain a proper oral hygiene. That is, we have to brush our teeth daily, twice, using a brush, a datun, or we can use dental floss. Not only this, we need to rinse our mouth after eating any kind of food, we have to also take care that we should not put our dirty hands into the mouth. Not only this, unwashed objects should also not be put into the mouth. Next is the tongue. Tongue is a highly flexible, fleshy organ, and it has many functions. That is, especially 3 important functions. It helps in speech, it helps to mix the food thoroughly, and also helps in the swallowing of the food. Not only that, it helps us to detect taste. We can detect different types of taste such as salty, sour, hot, bitter, etc. Because in the tongue are present taste receptors, which are known as gustatory receptors. They detect the taste, and we come to the understanding about what type of taste the food has. And it is said that we have different areas in the tongue for detecting the different types of taste. Now let us look into the salivary glands. That is the third part. Salivary glands, there are 3 pairs of salivary glands in the mouth. They produce a liquid called saliva, which helps in the breakdown of starch to sugar. Now, saliva contains an enzyme or a digestive juice called salivary amylase, and it is the salivary amylase that is responsible for the breaking down of starch to sugar. Let us now discuss an experiment to prove that salivary amylase help in the breaking down of starch to sugar. We need to take 2 test tubes and label them as A and B. In test tube A, we have to take boiled rice and 1 ml of water. In test tube B, we have to take rice that is chewed, and we have to keep on chewing this rice for 3 to 5 minutes so that it is thoroughly mixed with saliva, and then put it into the test tube B and add 1 ml of water. Later on, we have to put a few drops of iodine into both test tube A and B and keep this experimental setup as such. After some time, when we look at these 2 test tubes, we can see that in test tube A, the color has changed to blue-black. This is because of the presence of starch. While test tube B remains colorless because the salivary amylase that is present in the saliva breaks down starch to sugar. Therefore, there is no color change. Therefore, we can conclude that salivary amylase that is present in saliva helps in the breakdown of starch to sugar. With that, we have come to the end of today's session. Thank you.